In this 5-mark subjective question, they are directing us to understand the given systematic representation of the gene involved in the lac operon. With this specification, let me understand the given gene in this lactose metabolism controlling operon diagram to say the answer for the given sub-question. In the first sub-question, they are directing us to identify and name the regulatory gene in this operon, and then they are insisting us to specify in which way this operon is getting switched off. For this, we are specifying the I gene produces the repressor that binds to the operator. This happens in the absence of lactose or allolactose, thereby preventing the RNA polymerase interaction to the promoter. So the structural gene LAC Z, Y and A is not going for expression. For this specification, we are getting 2 marks. In the second sub-question, they are directing us to illustrate the negative regulation of the lac operon. For this, we are illustrating the repressor. Once it is getting interaction with the operator, the lac operon is getting switched off. For this specification, we are getting 1 mark. In the third sub-question, they are directing us to give the inducer molecular name and the gene product name of the gene Z and Y, and they are directing us to specify the gene product function. For this, we are illustrating the lactose or allolactose availability inactivates the repressor protein so that the RNA polymerase will interact with the promoter more effectively, and the structural gene LAC Z, Y and A will go for effective expression to make the enzyme beta galactosidase to facilitate the lactose catabolism so that we will be getting glucose and galactose for the cellular respiration. Whereas the polymerase enzyme, the outcome of the Y gene is facilitating the lactose interaction with the E. coli, and A is making the transacylase enzyme which is required for acylation process specifically for the lactose metabolism. In this way, if you are illustrating, we will be getting 2 marks to satisfy the 2 mark demand of the third sub-question demand, so that we can satisfy the overall 5 mark demand of this first optional subjective question. The second optional sub-question, they are directing us to specify the various genetic equilibrium factor in which way it is illustrated in the Hardy-Weinberg equation. For this, we are specifying the allele frequency in a population are stable and remain constant from one generation to another generation. So the sum total or the allele frequency is 1. For this specification, we are getting 2 marks, and we need to illustrate the gene migration, gene flow, genetic drift, mutation, gene recombination, natural selection leads to the distribution and disturbances in the equilibrium. For this specification, we are getting half mark. When changes in the allele frequency occurs many times in a population leads to the formation of a new species. For this illustration, we are getting another half mark. The original drifted population becomes the founder, and this effect is exactly indicating the founder's effect. For this illustration, we are getting another 2 mark. In this way, we are satisfying the overall 5 mark demand of the second optional subjective question.